Assalamualaikum. I, Dr. Muhammad Akram, Department of Eastern Medicine, Government College, University, Faisalabad. I was discussing the dehydrogenase. They are produced from different tissues, isoenzymes, LDH1, HH. It means tetra H, cardiac muscles only. LDH2, HHHM, LDH3, HHMM, LDH4, HMMM, LDH5, MMMM, skeletal muscles only. Isozymes of CPK, CPKMB in cardiac muscles, CPKBB in brain muscle tissues, CPKMM in skeletal muscles. Isozymes of alkaline phosphatase. It is present in bones, livers, placenta, and intestine. Lysozymes, lysozymes destroy the bacterial cell wall, thus provide protections against bacterial infection. These enzymes are present in saliva, tears, nasal secretions, and milk. Zymogens are pre-enzymes. Zymogens are inactive form of enzymes. They are the proteolytic enzymes which are secreted in their inactive form in order to protect the tissue that secrete them from autodigestion. They are subsequently converted to the active enzymes by removing the amino acids covering their active sites. For example, pepsinogen, which is a zymogen, is converted to its active form pepsin, first by ICL and then by pepsin itself. Pepsinogen is converted into pepsin in presence of ICL and pepsin. Another example of a zymogen is trypsinogen, which is converted to its active form trypsin, first by the enzyme enterokinase and then by trypsin itself. Trypsin also converts chymotrypsinogen to its active form chymotrypsin. Trypsinogen is converted to trypsin and trypsin and enterokinase is as act as enzymes. Chymotrypsinogens in presence of trypsin is converted to chymotrypsin. Conversion of zymogen by its own form is known as autocatalysis. Mechanism of actions of enzymes. The active site of the enzymes binds with the substrate and forms enzyme substrate complex, which is converted to enzyme product, which then splits into enzymes and products. Factor affecting enzyme activity, substrate concentration. The rate of enzyme catalyzed reactions increases with substrate concentration until the maximum velocity is reached. After reaching maximum velocity, the rate of the reactions become constant. Maximum velocity, it is defined as the maximum velocity of enzyme catalyzed reaction. This leveling of the reactions is at high substrate concentration reflects the enzyme saturation with substrate of all the available binding sites of are on the enzyme. Michaelis constant. According to the Michaelis-Menten equation, it is defined as the substrate concentration at which the reaction velocity is one half V maximum. It does not vary with the enzyme concentration. Low Km value reflects high affinity of the enzyme for the substrate. High Km value reflects low affinity of the enzyme for the substrate. Enzyme concentration. The velocity of enzyme catalyzed reaction is directly proportional to the concentration of enzyme. This is called the reaction of first order. Temperature. The velocity of reactions increases with temperature until a peak velocity is reached. This increase is due to the increased number of molecules having sufficient energy to pass over the energy barrier and form the products of the reactions. Further, elevation of temperature beyond the optimal temperature results in decrease in the reaction velocity as a result of temperature-induced denaturation of the enzymes. Optimal temperature. It is the temperature at which the enzyme activity is at its maximum. For human, it is 37 pH. Every enzyme requires specific pH for its maximum energy. Pepsin, a digestive enzyme in the stomach, is fully activated and pH 2, acidic pH, it will be denatured in an alkaline pH. Enzyme activity also depends on the ionic nature of the amino acids amino acid side chain, extreme of pH alter the ionic nature of amino acid side chain and thus halt the enzymatic activity. Products of reactions. If the products of reactions are removed as fast as they are formed, the reaction will proceed in the forward direction. If the products of reactions are not removed as fast as they are formed, then reaction will either slow down or will stop completely. Metallones. Many enzymes require metallones for their activity. Carbonic anhydrase loses its activity upon removal of zinc from the enzymes. Catalase requires both magnesium and zinc, and pyruvate kinase requires both magnesium and potassium. In the absence of an appropriate metal cofactor, the enzyme typically does not show its activity. Inhibitors. Enzyme activity is inhibited by the presence of certain substances called inhibitors. Inhibitors are inhibition of enzyme activity. There are two types: competitive and non-competitive inhibition. Competitive inhibition reversible. These inhibitors resemble the substrate. Inhibitors binds reversibly to the active sites which the substrate would normally occupy. Therefore, the inhibitors compete with the substrate for the active sites and block the formation of enzyme substrate complex. Dilution of the enzymes inhibitor complex by increasing the concentration of substrate will resume the enzyme activity. Non-competitive inhibitions irreversible. These inhibitors do not resemble the substrate. Inhibitors does not occupy the active site, rather it binds at some other site, like the site for cofactor or coenzymes to inhibit the enzyme activity. It results in the formation of three-dimensional structure of the active site, thus making it impossible for the enzymes to recognize the substrate and to make an enzyme substrate complex. Dilution of enzyme inhibitors complex by increasing the concentration of substrate will not reverse the enzyme activity. Thank you very much.